In this video, we're going to talk about a way that we can actually quantify evolution. So remember, evolution is the change in allele frequencies over time. And because you can count the number of alleles in a population, you can count and determine, calculate whether or not they have changed over a particular length of time. Now, our book calls this section, a trait does not decrease in frequency simply because it's recessive. And they're using this equation to show that natural selection or evolution itself does not always remove a trait simply because it's recessive. I prefer to think of this chapter as simply a way to quantify evolution and give you a quantitative value to say whether or not a population is changing. And so you can use something called the Hardy-Weinberg law and the Hardy-Weinberg equation to calculate allele frequencies at a given point in time and then calculate them again after a suspected evolutionary event and determine whether or not they have changed. Now, the basis of this law is that the frequency of alleles in a population always equals 1. So remember, a frequency is a decimal or a fraction. So all of the alleles in a population, if you calculate all of their frequencies, add them all up, they have to equal 1. So this is also sort of a good math check for you to make sure you're doing your problem right. So this image is from our white and yellow tigers that we talked about. I think it was in the second video. Now, P and Q stand for the frequencies of the dominant allele and the recessive allele, respectively. So let's say that Q is equal to our white allele and P is equal to the yellow allele. After an evolutionary event, Q is going to increase while the relative frequency of P decreases. Remember, they're always equal to 1. You only have a set number of individuals in a population, and even if that number changes, you can calculate the relative frequency of the alleles in that population. So continuing on, Q, or the recessive white allele, is going to increase while the other P or orange allele decreases. So this is something you're just going to have to memorize. We will do examples in class on how to actually calculate your allele frequencies once we get to lab and our classroom assignment. So the fun part, Hardy-Weinberg Equation. Please don't tell me you hate math. I'm sorry. Some people hate math. This is not hard math, and you will be fine. I promise this is not hard math. The hard thing that comes from this section is that students don't memorize some particular things about this equation. So now I've said the other dreaded word, the memorize word. You have to memorize things about math. So I need you to shut off your mental block about math for this section. I promise it's not hard math, but it will be hard if you don't recognize and memorize some things about this equation. So the Hardy-Weinberg equation says P squared plus 2PQ equals, plus Q squared equals 1. So if you look at what we have going on here, we have allele frequencies adding up to 1. Very similar to the previous equation. Now I know you're going to say, but there's squares and there's 2's and there's P's and Q's together. You just have to pay attention or mind your P's and Q's, okay? So P, we are going to say, is always equal to the dominant allele frequency. Now you calculate an allele frequency by counting the number of dominant alleles, dividing it by the total number of alleles. Q is going to be equal to the recessive allele frequency. Again, you calculate this by counting the number of recessive alleles, dividing it by the total number of alleles. Keeping in mind that all individuals have 2 alleles, the same or different, for each trait. So the number of total alleles is going to be 2 times the number of individuals in your population. Again, we will practice this. Now, you have to remember those things. You have to remember a couple of more things. So, the probability of getting two dominant alleles is just the probability of getting one allele times the probability of getting the other allele. In other words, p squared. Probability, or p squared, is going to be the dominant allele frequency squared. Now this, because you are getting two copies of the same allele, this is the same thing as the frequency of the homozygous dominant individuals. Q squared is the opposite. The probability of getting one Q times the probability of getting the second Q is going to be your frequency of homozygous recessive individuals. And then finally 2PQ, that one in the middle, this is your heterozygous individual. Now remember, you could get a white allele from mom, an orange allele from dad, or you can get the opposite. You can get the orange allele from mom and the white allele from dad. So there's two ways that you can be P and Q together. So 2 times the frequency of P times the frequency of Q. That's going to tell you how many heterozygote individuals you have in your population. Now we can use this to determine how many individuals we expect to have the dominant phenotype or the recessive phenotype. So the other thing we can use this equation for is to determine how many individuals we think are going to be homozygous recessive, or the frequency of homozygous recessive individuals. So not only is Q squared the frequency of the recessive genotype, it's also the frequency of the recessive phenotype. That's another thing you're going to have to memorize. This P squared plus 2PQ, if you think about what that is, that's the dominant phenotype and the heterozygous but dominant phenotype. So 2PQ, sorry I said that backwards, P squared plus 2PQ is going to be the total number of individuals that have the dominant phenotype. So you have to remember that as well. You really have to memorize these, and you have to understand what all of these words mean. You have to think about the relationship between these numbers and the genotypes and the phenotypes. Once you get that straight, the math is the easy part. So why do we use this Hardy-Weinberg equation? So we use this because if we know the allele frequency in a population, we can predict the genotypes and the phenotypes that we would expect to see. Then we can relate that to evolution. If the phenotypes that we see are not what we predict, then the population is evolving. In other words, they are outside of something called Hardy-Weinberg Equilibrium. If the population is not evolving, meaning that allele frequencies are not changing, the population is said to be in Hardy-Weinberg Equilibrium. As long as a few Hardy-Weinberg assumptions are not violated, evolution does not occur. So in essence, this is saying that as long as those four mechanisms of evolution are not taking place, our allele frequencies are not going to change from generation to generation. So let's look at how we actually use this Hardy-Weinberg equation. So let's say we have a population of 1,000 kangaroo rats, and of that 1,000, we have dark brown, spotted, or light brown kangaroo rats. So the dark brown is the dominant trait, light brown is recessive, and in this case we have the spotted, which is actually a incomplete or codominant situation. So they have given us the number of individuals. So we have 16 individuals who are dark brown, 222 that are spotted, and 762 that are light brown. So we can see that recessive allele is actually the most frequent in the population. So just because it's recessive doesn't mean that it is going to be lost from the population or become less frequent. So let's calculate the frequency of alleles. Now I'm going to point out right here, this is the number of P alleles divided by the total number of alleles. And we have 1,000 kangaroo rats Each individual has 2 alleles. So our total number of alleles is 2,000. Now we need to calculate the total number of our dominant allele. Remember, the frequency of the dominant allele is equal to P. So we have 16 individuals who are dark brown. 16 individuals times 2 alleles each. So each dominant allele has 2 copies of B. That's why we multiply this one by 2. We also have these kangaroo rats that have 1 B allele. So if there are 422 individuals and they each have 2 alleles, that means there are 222 B alleles and 222 little b alleles. We're going to use the 222 big B alleles as part of our calculation. So we have 2 times 16 for 2 copies of big B plus 222 for 1 copy of big B divided by 2,000 total alleles, giving us an allele frequency for the dominant allele of 0.127. Now, we can calculate Q 2 different ways. Remember, P plus Q equals 1. So at this point, we can use that equation, take 1 minus 0.127 to get the frequency of Q. Instead, we're going to walk through this calculation again. On paper, I don't care which one you use unless I specifically ask you to directly calculate both. So again, to calculate the recessive allele, Q is equal to the frequency of the recessive allele, we're going to start with our 2,000 alleles. Remember, that's because there are 1,000 kangaroo rats, 2 alleles each. Now, we're going to calculate the total number of little b alleles. So there are 762 individuals. They each have 2 copies of the little b allele. So we have 762 times 2 copies each, plus those alleles that are in the spotted kangaroo. Remember, there are 222 individuals. They each have 1 copy of big B, 1 copy of little b. So we have to add in our 222. So the frequency of our light brown or our recessive allele is equal to 2 times 762 plus the 222 from that spotted kangaroo rat divided by 2,000 total individuals gives, 2,000 total alleles gives you a frequency of the recessive allele at 0.873. So you can see that the frequency of the recessive allele is significantly greater than the dominant allele. Now, let's calculate the genotype frequencies for this allele. So this diagram is really helpful. It just shows you where you get the p squared. So the frequency of the big B allele is 0.127. Frequency of the big B allele from the other parent, 0.127. That's equal to p squared, which is 0.016. So that is the frequency of our homozygous dominant genotypes. Q squared is the same thing. Chance of getting little b times chance of getting little b is 0.762. And then the frequency of the heterozygous genotype is 2 times PQ. So the probability of getting little b, probability of big B, and we can get that in 2 different ways. So 2PQ is equal to 0.222. Now, what does this have to do with evolution? So assuming that there is random mating, which means there's no selection for this particular allele, and assuming that there's no mutation, no gene flow, no genetic drift, and no natural selection. So in other words, assuming no evolution is occurring, there should be no change in allele frequencies in the next generation. So we can check, has our genotype frequencies changed? No, they haven't. You can see that they're the same as the previous population. That means that there is no evolution occurring in this population. So let's talk about Hardy-Weinberg equilibrium and what this means. Hardy-Weinberg equilibrium says that the population is not evolving. The population does not evolve if there's no natural selection, no mutations, no migration, no genetic drift, and that there's random mating, meaning there's no sexual selection happening. If a population is in Hardy-Weinberg equilibrium, there is no evolution occurring. So if a population is in Hardy-Weinberg equilibrium, we say that none of these mechanisms of evolution are happening. It is very likely that I will ask you to list the Hardy-Weinberg equilibrium conditions. It is also important that you state that in equilibrium there is no natural selection. If you don't have the no, your question is going to be wrong, because Hardy-Weinberg equilibrium says that none of these conditions are being violated. If any of these conditions are being violated, then evolution is going to occur and allele frequencies are going to change. And I wanted to make a note about a couple of these. We all know what all the top 4 are. Genetic drift is also sometimes called infinitely large population. And this is because in a large population, the changes in allele due to chance are very likely not going to have a large effect. In a small population, they can have a major effect. So depending on the book, sometimes it's called a large population. And so if you have a small population, you're assuming that genetic drift is going to have a big impact on that population. Random mating basically means no sexual selection. It also means that you're not being forced to mate with a close relative because of small population size. So random mating is essentially changes in genotype frequencies without changes in allele frequencies. So they're changes in a population without evolution occurring, which is sort of a special case. So let's go through another example. This example is using Pku, which is a condition that we talked very briefly about in our genetics section, and it is the inability of individuals to process phenylalanine in the diet. That's an amino acid. So you can use Hardy-Weinberg to determine whether evolution is occurring. You can also use it for another reason. You can also use it to determine how many individuals you expect to have a particular disease, in this case Pku, which is a recessive. So you can figure out the frequency of individuals with the disease, or you can figure out the percentage of carriers of that disease. So you can use this information to determine phenotype or genotype frequencies for other purposes as well. So the rate of occurrence of Pku is 1 in 10,000 US births. So we're going to start there. Those are individuals who have the Pku phenotype. So if the if you know the number of individuals with Pku, you automatically know a couple of things. You know the frequency of individuals with the homozygous recessive genotype, because Pku is a recessive disorder. Now that means that you know Q squared, because Q squared is the frequency of homozygous recessive individuals. It's a frequency of the homozygous phenotype, and it's a frequency of the homozygous genotype. So you can use that information. You can say 1 in 10,000 people has the homozygous recessive genotype. Therefore, 1 in 10,000 people is equal to Q squared. So if we do this math, Q squared is equal to 0.0001. Take the square root. Q is equal to 0.01. So this is your frequency of the recessive allele in the population. Now, we cannot calculate 2pq plus p squared and use that to get p. The reason is that we don't know how many individuals are heterozygous. So you cannot take the opposite problem and say, okay, I know how many dominant phenotypes we have and therefore calculate p squared. It doesn't work because you can't tell how many Qs are present in the heterozygous individual. So keep that in mind, because I guarantee that question will come up later. It's one of my favorite ways to stump students on this question. So because of that, we have to use the fact that p plus q equals 1 in order to calculate p. So if p plus q equals 1, Q equals 0.01, then the frequency of our dominant allele is 0.99. But our ultimate question is, how many people are carriers for this disease in the United States? So we can use our newly calculated P and Q values to determine the number of heterozygous individuals. Now remember, the 2PQ part of the equation is going to tell us our frequency of heterozygotes, or our frequency of carriers in a population. So 2PQ is equal to 2 times our P value, 0.99, times our Q value, 0.01. So 2PQ is equal to 0.0198, ... which is approximately 2% of the population are carriers for PKU.